What does DNA stand for? The letters DNA in medicine stands for deoxyribonucleic acid. DNA is a complex and fascinating molecule that is essential for life. All living things have DNA, and it is the genetic material that contains the instructions for the function and growth of cells. It is like a kind of recipe for life, determining how cells should be, how they should function, and how they should reproduce. What is DNA? DNA is a long, thin molecule composed of two complementary helices that wind together like a step. Each helix is made up of a sequence of basic units called nucleotides. There are 4 different types of nucleotides in DNA: adenine, cytosine, guanine, and thymine. The sequence of these nucleotides is what determines the genetic information. DNA is found in the nucleus of every cell and is protected by the nuclear envelope. When a cell divides, DNA is replicated so that each daughter cell has a complete copy. DNA function. But how is DNA information used to control cell function and growth? The answer is through gene expression. Gene expression is the process by which DNA is read and used to make proteins, which are molecules essential for life. Proteins are responsible for many important functions in the body, such as growth, tissue repair, and immune response. Each protein is task-specific and is produced from a specific gene in DNA. DNA and evolution. In addition, DNA is also important for evolution. Genetic variation is the basis of evolution, and DNA is what enables variability. Mutations in DNA can produce new characteristics in cells, and through natural selection, fitter cells survive and reproduce, resulting in an increasingly adapted population of cells. Another interesting aspect of DNA is that it is possible to use it to trace the evolutionary history of living things and their phylogenetic relationship. The DNA sequence is unique to each species and can be compared to determine similarities. DNA replication. In molecular biology, DNA replication is the biological process of producing 2 identical replicas of DNA from one original DNA molecule. DNA replication occurs in all living organisms, acting as the most essential part for biological inheritance. This is essential for cell division during growth and repair of damaged tissues, while it also ensures that each of the new cells receives its own copy of the DNA. The cell possesses the distinctive property of division, which makes replication of DNA essential. DNA is made up of a double helix of 2 complementary strands. The double helix describes the appearance of a double-stranded DNA, which is thus composed of 2 linear strands that run opposite to each other and twist together to form. During replication, these strands are separated. Each strand of the original DNA molecule then serves as a template for the production of its counterpart, a process referred to as semi-conservative replication. As a result of semi-conservative replication, the new helix will be composed of an original DNA strand as well as a newly synthesized strand. Cellular proofreading and error-checking mechanisms ensure near-perfect fidelity for DNA replication.